if you're interested, as well as the one here. This one here is the one that's supported by the same groups that are supporting the summer school here. And again, this is designed to go ahead and provide pre-doctoral fellows as well as postdoctoral fellows research opportunities. And we have more than 20 laboratories here at the University of Colorado that participate in this program. I want to just highlight the program leadership here. We have Josie Broussard, who's a professor at Colorado State University. Stacy Simon is a professor at our University of Colorado Anschutz Medical Campus. Professor Mark Opp, who's here at CU Boulder, and you'll hear from him today. Professor Emma Lanson, who's also at our Anschutz Medical Campus, and myself form the executive committee for this particular program. And so it is multi-institutional, is also important to note. And as I mentioned, we have more than 20 laboratories that are conducting sleep and circadian science and related transdisciplinary science in this space, covering a variety of topics. So as you'll see from this brief list here, there's a lot of different types of research opportunities and research training opportunities here. And I'll note that our applications for 2026 are going to be due in January of 2026 for those who are interested. And you can reach out to me if you have any questions. We also have, as I mentioned, an undergraduate research program, and this is designed to promote careers in sleep and circadian science and introduce people to this area and integrate it with other types of science that they're doing. And this one's also sponsored by the National Institutes of Health. And in this case here, it's designed to help prepare people for entering into PhD and MD, PhD type programs and studying sleep and circadian neuroscience and sleep and circadian science broadly. And applications for summer of 2026 are going to be due in early 2026. And in terms of the leadership there, this is a multi-institutional effort, as I'll mention here in a moment. And the leadership team here is Professor Joe Takahashi, who's at University of Texas Southwestern, Professor Gianluca Tosini, who's at Morehouse School of Medicine, and myself, as well as Dr. Christina Alston, who's here at CU Boulder, and Professor Anita Shelgakar, who's at the University of Michigan. And this particular training program, as I mentioned, is designed to introduce students, and you're going to meet some of our R25 students who are here in the audience here today. They'll be presenting their science at the end of this week. And this is, again, multi-institutional, so different institutions train these R25 scholars every summer. And so these are the previous years. This year, we had training done at Harvard, Michigan, Stanford, and Colorado. And next summer, we're expecting Penn, Washington University, St. Louis, and University of Texas Southwestern. All right. So this particular summer school, a little bit of quick history here. We first were going to start it, and then COVID hit. And so we had to pivot immediately. And so we had our first two summer schools virtual. And we were able to reach many people around the world. And it was so successful that we decided that we're going to continue that. All right, and so that's why this is hybrid. It's not just in person. All right, and so we're going to see that we're going to have some advanced training in person, some of the workshops, and some of the discussion groups here. So for those who are in the virtual audience or are interested in that in future years, please do come join us in Colorado. But we're going to go ahead and have a lot of our talks available, key talks available virtually. And this is 1 week here at the University of Colorado Boulder. And in general, in the morning, we're going to have advanced intensive discussions, lectures, that type of thing. In the afternoons, we typically have workshops and professional development activities as a general rule. We do have, again, a smaller group here in person this year, just around 50 people. And there's an application process for this. So for those who are in the virtual audience, maybe interested next year, please do look out for that on our webpage. This course is made free, so we don't charge for it. And the only thing is if you're coming to here in person, you got to take care of your own travel and housing and food. And that's what the trainees or their mentor are responsible for in person. Now, I'm excited to say this year we've got 35 countries participating with us here. You can see they're from all over the world here and from 16 different United Nations sub-regions around the world here. And so everyone who are participating from around the world, welcome. And for those who are in your wee hours of the night there, where we are here, welcome for as much as you can stay. or we'll see you again when you wake up in the morning. All right. So, program. After this introductory lecture here, we're going to have our first plenary by Professor Mark Opp, who is again here, professor at the University of Colorado Boulder. And he's going to be talking to us about historical aspects of sleep and the immune function. And he has got a catchier title that you'll see here in a moment. And then we'll have Professor Till Runenberg from the University of Max Planck University in University of Munich in Munich. He'll be talking to us about the history of circadian science from the Rothbard to humans. And again, I think most of the faculty are changing their titles, so I'm sure he'll switch his around too. And then, so those two are going to be virtual. Then we'll have a pause, and we're going to have a data blitz for in person here only. And these data blitzes are designed to go ahead and allow the trainees who are here at the summer school, who are going to be presenting their research and posters, to go ahead and give us a brief snippet of what they're going to be talking about, so that way we can all know what we're going to see later this evening in the poster session, which again is in person only. Then we'll break for our lunch. And then in this case here today, this is the first day, we are going to have two more talks that are going to be virtual. And so we'll have Professor Elizabeth Klerman coming from Harvard Medical School, and she'll be talking about adding sleep and circadian rhythms to medicine. And then we'll have a coffee break here, and then I'll give a brief talk about circadian biomarkers. And then we'll have our first poster session. So again, Professor Ops, Ronenberg's, Klerman's, and then my talk will all be virtual today. Then tomorrow we'll have Professor Danny Forger from the University of Michigan starting us off in the morning in the virtual sessions. He'll be talking about wearable technologies for sleep and circadian science in real life. And then we'll have Professor Bryce McConnell coming from our University of Colorado Anschutz Medical Campus talking about sleep EEG and cognitive decline, and using the EEG as a biomarker for aging and age-related disorders. And then we'll have our second data blitz again, which will be in person only. And then the rest of our Tuesday is all going to be in person. So this will not be available virtually. So we'll just have these first two talks available virtually on Tuesday. On Wednesday, again, we'll have two other additional virtual talks. We'll have Professor Paul Shaw from Washington University St. Louis talking about genetics of sleep in animals and humans. And then Dr. Rob Thompson, who's going to be talking to us, who's here from the University of Colorado Boulder, talking to us about the microbiome countermeasures to improve sleep and circadian rhythms. And then with the in-person summer school, we'd like to go ahead and have a little brain break in the midweek here. And so the rest of the day on Wednesday will be free time. On Thursday, we'll have additional lectures, and we're going to have 3 virtual sessions combined, so hybrid sessions. So Dr. Zoe Panachal, she's going to be talking to us about the state of the science on sleep and cannabis. Then we'll have Dr. Stephen Duntley, from our medical school also, who's going to be talking to us about clinical sleepiness and fatigue. After a lunch break, we're going to have an afternoon advocacy workshop. And so we're going to have Professors Janet Mullington from Harvard Medical School and Beth Israel Hospital, Professor Alan Pack from the University of Pennsylvania, and Professor Fred Turek from Northwestern University joining us virtually for a discussion about advocacy of sleep and circadian science. And then we will have an afternoon workshop that is not virtual. So this one here, this advocacy, will be virtual. And then on Friday, which is our last day, we'll start off the day with a talk by Professor Rachel Rao, who's here at the University of Colorado Boulder, talking about sleep and traumatic brain injury. And then that will conclude the morning session for our virtual component. We'll have a workshop that's in person only. And then in the afternoon, we're going to have virtual hybrid presentations from our R25 scholars, where they'll be talking about the research, the science that they did this summer on the program, and we'll get to all hear about their work. All right, so that is the actual structure of the summer school here for this week. A little other business here. For those who are participating virtually, we are going to use the Q&A function here for the webinar. And so you can ask questions as we go. And to the audience also, you can ask questions of our faculty here as we're going throughout their talk, right? So we don't have to wait to the very end. The only thing that I ask, and we're going to follow what I've come to know is called the Beth Clerman rule. Dr. Clerman reminded me that anytime you ask a question, to please state your name and where you're from so people get to know you and who you are, right? And so in terms of the virtual audience, you can do anonymous if you want, or you can go ahead and have your name and location in the question itself. But
there's also for doing a comment,
okay. And Professor Ronenberg just said it also goes for making comments. Thank you, Professor Ronenberg. I will be monitoring the Q&A live. And so if we do get questions that come in, I'll interject them as we go. All right. All right. And then if there's anything else, if there's any questions that come up during the week, please feel free to email me and I will get to back to your email as soon as possible. All right. So with that, I'm going to switch slides here and we'll have our first lecture of today. So this works here. You can see, unless you want the pointer. But the
laser might show up.
laser. Let me see the laser here. Perfect.
Yep. Wait, no, it was working with the notes. Who? Okay.
All right. So that's right.
Well, thank you for that kind introduction. No, it's a pleasure to be here, and I'm glad that you all can join us from near and afar. As Ken said, this is the 6th annual summer school, and it's always been a pleasure for me to participate at some level. Now, Ken asked me if I would be willing to give a history of how we got to the place where we are now in the context of sleep and immune health. So, a spoiler alert, as we go through this historical narrative, it is a story, but it's intertwined with a lot of personal interactions. And because I happen to be training in the early days of the development of this field in the lab that was at the forefront, there's a little bit of egocentricity in my presentation today. But I feel I can get away with it because I just retired and went emeritus, and now I can do whatever I want without fear of repercussion. So with that, since it's a historical narrative, we have to start with a quiz, and hopefully this will also serve as an icebreaker. So we're going to start with some questions. The first question is, what discovery in 1953 changed the field of sleep? And I'm looking for real answers here, not silence. Anybody? Yeah.
Circadian rhythm is longer than 24 hours.
I'm sorry.
Daryl from UMBC, and the circadian rhythm is longer than 24 hours.
Not the answer I'm looking for. Anyone else? And this is why we need a lecture on history, right? Azarinsky and Kleitman described eye movements during sleep. So what do we now know this stage of sleep? REM sleep. Okay. Next question. We're not doing very good so far, so let's pick it up a little bit here. If my slide will advance. Who first recorded the human EEG? And this is multiple choice, so I'm making it easier for you. Von Economo, Berger, Kleitman, or Jouvet. So those of you who think the answer is A, von Economo, raise your hands, please. Those who think it's B, a few. Faculty, keep your hands down. Kleitman, any responses for Kleitman? All right, a few. And Jouvet. All right, so the vast majority of you are chicken and didn't even put your hand up. So the answer is Hans Berger. Now, he actually, we'll talk about Berger a little bit more later on. He actually used methodologies that were developed in the latter part of the 19th century, but surprisingly enough, was the first to record the human EEG in 1924, although he didn't publish the paper till 1929, and I'm not sure why that happened. Okay, next question. Who wrote the first clinical sleep scoring manual? Again, multiple choice to make it easy. Those of you who think it was Dement and Rothward, letter A. One hand, two hands. Chase and Harper. No hands. Sorry, I don't know what you guys virtually are doing. You can raise your hand if you want, but since you're probably by yourself, it won't matter. So, letter C, Rechtschaffen and Kales. Okay, quite a few more hands. McCarley and Hobson. Maybe a half a hand. All right. The correct answer is, of course, Rechtschaffen and Kales. If you ever have an interest in anecdotes about Al Rechtschaffen, you should talk to Paul Shaw. Paul trained with him and has lots of interesting stories to tell about one of the founding fathers of our field. I put this picture of Dr. Anthony or Tony Kales because from their family perspective, they've had a very interesting impact on sleep. So both Tony and his wife Joyce were PhD investigators focused on sleep research. This is their daughter Helen at age 6, who was not only the subject of many of Tony Kale's studies, but also went on to become a PhD and study sleep. So there's a real family history here of involvement of sleep. Okay, last question, I believe. Who started the first sleep disorder center? Dement, Chase, Rechtschaffen, or McCarley? All hands for Bill Dement. Okay, a few. Michael Chase. None here. Alan Rechtschaffen. 3 or 4. Bob McCarley. 1. All right. The correct answer is Bill Dement. So here's a picture of Bill in his kind of mid-career just a few years ago. And now I want to ask a bonus question. What is the importance of the picture with this dog, and what the heck is going on with that dog? Yes.
Older. The dog is experiencing cataplexy, which is a symptom of narcolepsy, and Dement was one of the first to create a colony of dogs to discover the mechanism behind narcolepsy.
Perfect. Great answer. You go to the head of the class. Thank you so much. Bonus question number 2. Where did this occur? Where did he establish his sleep disorder center? Now, you had your chance. Anybody? It's west of the Mississippi, west of the Rocky Mountains. I'm not seeing a hand. Oh, way over. Yes.
I would say Stanford.
You want to be correct. So thank you very much. All right. So the next question I want to focus on, and actually would like a little bit of discussion because I'm not sure I know all the, in fact, I'm positive I don't know all the answers, is why is it important for us to understand the history of sleep research? You know, why did Ken ask me to give this lecture? Why do I have an interest in giving this lecture? So I have a few ideas here, but I also want to hear from you why you think it's important that we understand something about how our fields developed. And later we're going to hear a similar time course of development within the circadian field. So if we take 1953 and the discovery of REM sleep or rapid eye movements as kind of an index point, from that period, Ken and Paul and Till and Beth, we're probably generally the third generation of sleep researchers, academically speaking. So those of you in the audience who are currently training would likely be the fourth generation. What this means, and the fields of sleep and circadian research evolve generally in parallel. So what this means is that now our respective fields are mature, right? We're established. We're no longer having to fight some of the battles that we did when these nascent research areas were developing. With maturity comes a couple of positive aspects and some negative ones. With a mature field, it's much easier to draw individuals and investigators from other fields into this domain that we would generically call sleep or circadian research. That also means that these incoming investigators, which can contribute to the field, often have not any idea how we got to the place where we are today. Paul and Ken and other faculty here, we trained with founders in the field, the founding fathers. That connection isn't available for people who come in later as the discipline matures. So within that context, I'm always been fascinated with history because of the almost anchor that it gives me in this area. I know these individuals who made the initial discoveries that really shaped our field. And that's important to me. And I hope by the end of the presentation, you'll share some of my enthusiasm for history. Now, there will be data slides, but the data that I'm going to share with you is presented within the context of the historical narrative that we're talking about. Keep in mind that the narrative that we want to address, specifically for me, is sleep and immune health. The early history of the field wasn't necessarily focused on sleep and neuroimmunology or sleep and immune interactions, but eventually we got to the place with discoveries that were important that we could no longer ignore the immune system within this developing story. So that's where we're going to go. I'm going to start with some current statements that as of maybe only 25 years ago or 30 years ago, if you made this statement, you would be hard to back it up with empirical evidence. And that is that chronic insufficient sleep kills. Now, this is almost a truism in the fact that it's widely accepted that chronic insufficient sleep is associated with negative health outcomes and can drive pathologies to the point of death. And just as 3 examples, these are 3 big data studies from among many that I could choose that demonstrate these strong associations between chronic insufficient sleep and various aspects of health and disease. And if we take a look at meta-analyses or systematic reviews, there are an incredible number of disease states and pathologies that are associated with chronic insufficient sleep. If you are interested in medical school or you're a physician already, this is like a smorgasbord of pathologies to choose from, and almost every discipline in medicine is represented by an impact or an association with insufficient sleep. So we're to the point now where there is an understanding that interactions between sleep and the immune system are bidirectional, right? If we have an immune challenge of some sort, or we're subject to a chronic pathology, our sleep is disrupted. Conversely, chronic insufficient sleep or disrupted sleep, even in healthy organisms, can drive inflammatory processes and either exacerbate existing pathologies or actually induce a pathologic state. So it's now well accepted in the field that these are reciprocal interactions, and these 2 systems that for a long time were felt to be independent actually talk to each other a lot. All right, and just as one example, if we think of cardiovascular disease as the #1 killer in the US and Europe, which it is, recently the American Heart Association added sleep to their lifestyle parameters that affect cardiovascular health. And they actually have started a kind of a PR campaign that's focused on sleep. 15, 20, 25 years ago, this would have been unheard of. So this is the maturity of the field about which I spoke earlier. And now we want to move into some of the history as to how we got to the point where we can talk about these kinds of interactions and the importance of sleep to immune health, which affects almost every system in the body. So at the end, I'm going to put up a QR code from a manuscript that's published that contains this timeline. So don't worry about taking a lot of notes or anything now. You'll have access to it later. We will speak about various aspects of this timeline, but it's just to give you an overview of where we start and where we end up in the 2010s from a historical perspective. So I'm going to break this down into sort of almost quantile time periods, but of course you understand that history is fluid and it's often difficult to determine where one phase might end and another begins. So we know that at least at the time of the ancients, particularly Aristotle was writing about sleep and waking and actually wrote accounts of the common feeling of altered sleep during infectious challenge. He was specifically talking about malaise and how when we're sick, we often feel lethargic and we suffer from this malaise. And we don't have time to go everything from, Aristotle to present. So we're going to leave that for now. We're going to skip ahead to the latter part of the 19th century, which really set the stage and it serves as the foundation for much of what we do in sleep. And this was particularly the idea that there were fatigue substances, and we'll talk about sleep factors in a few minutes. But it was really the discovery of a couple of substances that set the stage for the developing field of endocrinology. These were the discoveries of renin and secretin. And surprisingly enough, as the field of endocrinology developed, neuroimmunology, which is a more modern term, actually evolved from the field of endocrinology. So we'll get there eventually. I want to start, though, the historical narrative with respect to sleep and immune function with some of the early sleep deprivation studies. This is an image of Maria Manasina. She was a Russian physiologist and doctor. She is thought to be one of the very first women to go to medical school and obtain her medical degree in Russia. She was very much interested in sleep health. And as an aside, she wrote an amazing textbook that was published in 1897 or translated to English in 1897 that probably serves as the first comprehensive sleep medicine text. In her early studies, if you want to understand the importance of something or the function of something, as we'll hear later, one way to address that question is to deprive the organism of that process and see what happens. So sleep deprivation in the context of our field is the most frequently used tool to address function. These sleep deprivation studies systematically started with Manasina in the late 19th century. What she actually did, we would not be allowed to do today, but she kept puppies awake and noted that if they were kept awake by not allowing them to go to sleep or they had to remain active, they would die in about 4 or 5 days. As a kind of control experiment, she deprived puppies of food, and after 20 or 25 days, they could still rescue the animal simply by providing them food. So she concluded that sleep was more important than food in a physiologic context, and that she actually made observations that were lost for decades. She observed that there was brain activity, that the brain was not silent during sleep, but none of these studies were actually a focus of investigation until the 1930s. In other studies, and here's where we begin to see some impact on the immune system, during this deprivation process, she noted that the body temperature of the puppies would drop as they progressed through this chronic sleep deprivation. Red blood cell numbers were reduced. There were hemorrhages that could be identified in brain, and some of these cerebral ganglion were impaired in their function. So these were the first systematic sleep deprivation studies, at least of which I'm aware. And I haven't been able to find any documented studies done before this time. So not directly related to sleep and immune function, but important for a lot of work that's been done in the sleep and circadian fields, were the first sleep deprivation studies, systematic studies done in humans by Patrick and Gilbert at the University of Iowa. Basically, in a nutshell, they demonstrated that with increasing periods of wakefulness, performance, which was basically reaction times measured by this instrument, decreased. So they were the first to demonstrate that sleep deprivation of humans impairs performance. That's just a aside because these studies were occurring about the same time as Manasina did hers. So if we skip ahead 10 years or so, two independent laboratories, one in Japan by Professor Ishimori and another one in France by Henri Perron and Legendre, and we'll get to them on the next slide, apparently ran across reports of these studies by Manasina and decided to investigate what it was that would cause animals to die. They were more interested in the sleep aspects as well. So the process that they used was somewhat similar to Manasina, but the outcome measures were different. So they deprived dogs of sleep, and then they would extract either cerebral spinal fluid or in some cases brain homogenates from those animals and inject those substances from the sleep deprived dogs into rested recipient animals. And when they did that, they observed that the recipient animals, even though they were well rested, would fall into a deep state of sleep after administration of these materials from the sleep deprived animals. So this led Ishimori to make perhaps the first statement that ever existed about the hypnotoxin theories of sleep. Something accumulates during prior wakefulness that triggers subsequent sleep. So Pierron in France did a similar types of studies, and their papers are published relatively close together in terms of years, but came to the same conclusion that a hypnotoxin accumulates during waking, which subsequently produces an increasing need for sleep. So these are the first sort of modern statements that suggest that there are sleep factors or hypnotoxins or hypnogenic substances in the brain. Now, it was, as you'll see, going to be a substantial period of time before we actually knew what some of those substances were. But these were the studies that set the stage for now our modern investigations. So if we jump ahead now to the 1950s, we have to pay homage to the neuroanatomists of the time. The period from the 1920s to the 1950s in neuroscience in general and in the field of sleep, which was very young at that time, was really dominated by neuroanatomic studies and neuroanatomists. The techniques that were being developed and used were focused on determining which brain regions, which brain nuclei were involved in the regulation of sleep and many other processes, but we're focused on sleep today. So there were many classic studies done by a variety of neuroanatomists in different locations around the world that helped us understand where in brain sleep is regulated and or modulated. So not on this list with Hess, Nauta, Ransom, Moruzzi, and Magoon is perhaps one of the better known. And this is why I'm going to ask you another quiz question. Which neuroanatomist first postulated that there were sleep and wake centers in the brain? Von Economo, Berger, Hess, or Nauta? All those who think von Economo, 2, 3.5, or 4. Berger, more hands. Hess, a couple. Nauta, one in the back, I think, just to be polite, because everybody deserves a vote, right? All right, the correct answer is von Economo. Von Economo was a neurologist and psychiatrist working in Austria, and many of his patients had been through what is colloquially called the Spanish flu, but probably wasn't, the pathologies that he saw probably weren't directly related to the Spanish flu. He focused a lot of effort on patients with encephalitis lethargica. And what he noted, without going into any greater detail, is that his subjects that had lesions in the anterior hypothalamus often complained or were insomniac. So in the anterior hypothalamus, if that area was lesioned, then individuals were more awake than they should be. So he postulated there must be a sleep center there because if it's gone, you're awake. Conversely, subjects with lesions in the posterior hypothalamus most often had extended periods of insomnia. So by using the same logic, there must be a wake center there. So he is the first individual to posit that there are discrete brain regions that are involved in the regulation of sleep and wakefulness, and that they are in different parts of the brain, in this case, the hypothalamus. Okay, now we're back to Hans Berger for just a little bit to remind us that he was the first to record human electrical brain activity. Catton had recorded electrical activity in the latter part of the 19th century on animals, but nobody had done this on humans. And it's an interesting story here. Berger had a subject who had had repeated operations to try and remove tumors that were causing problems. They weren't trying to localize epicenters for epilepsy exactly, but he'd had multiple surgeries to try and address or get this tumor out. As a consequence, he had a hole in his skull. So all Berger did was the same thing that Catton did with animals. He placed electrode bare leads on the surface of the brain and recorded electrical activity. So in the 30s, we saw a lot of studies focused on determining changes in brain electrical activity through the course of the night. I'm going to show you a few slides from Blake and Gerard's paper in 1937, which is seminal, but there were a lot of papers by other investigators as well. Loomis and Harvey and others published a lot of papers describing changes in EEG or electrical activity through the course of the night. I want to draw your attention to a few of these features within the context of the developing field of sleep. So they knew by 1937 that there were different waveforms that could be detected. And if you notice, these waveforms differ in their amplitude, you know, how much voltage is expressed peak to peak, and in their frequency, how many cycles in 1 second are manifest. So you can obviously see that there are a big difference between the waveform in line 3 versus that of line 1. So they already knew that there were different waveforms. They'd already demonstrated that during sleep, Noise could alter the EEG and induce an arousal, and in this case, for this subject, an airplane flew overhead. Here's another example of the type of waveforms that they could record, but I want you to focus on this one. So the bottom trace, I know it's difficult to see because it's an old paper, is from an individual who's awake. And the top figure is from the same individual 40 seconds later. So notice the difference between the low amplitude, high frequency EEG when the individual's awake, and this extremely high amplitude and very slow frequency EEG 40 seconds later. So unless you cheated and read the caption, what do you think could account for this kind of change in EEG within less than a minute in this one subject? Any ideas what's going on here? Okay, this subject had been sleep deprived for 60 hours and then allowed to subsequently sleep. So in the matter of seconds from waking to sleep, we now can observe what became to be called, for a while anyway, supranormal slow waves. Now we call this delta wave activity or other names, but early on, these were supranormal slow waves. All right, this is the last figure I want to show you from this paper because it's important for reasons I hope you'll understand shortly. Now, I've annotated the trace or the figures with the descriptions from the figure caption. So these are not my words. These are the words that Blake and Gerard used. Now, what is represented here are samples of an EEG from the same subject starting at midnight and going to 2:30 in the morning. So samples of EEG across a 2.5 hour period in the middle of the night. And as you can see that in 1937, they were already able to distinguish associations of distinct wave patterns in the EEG with some behavioral outcome measure, because they use expressions like deeply asleep, falling asleep, fairly deeply asleep. Remember, in these early days, we didn't have any standard scoring manual to tell us how to talk about what they were observing. Drowsy, etc. So the point I want to make is in 1937, it was already clear that through the course of the night, different stuff was happening in the brain because you got all these different transitions from one waveform to another. So this is 1937. When was REM sleep described? Can you remember back to one of our quiz questions? What year? 53? So that to me sounds like 15 years. So what always amazes me is how, for many, many investigators working in this area, the data are right here. There's something different. It's not a unitary state during sleep. But for 15 years, it was like apparently staring at the forest and not seeing the trees. So I find that fascinating, that everything that should have told us that different stages of sleep existed was there, but it took Azarinsky and Kleitman to kind of pull it together. Why? Because they looked at something that nobody else had looked at, which was eye movements. Okay, so now any questions or comments about this first sort of early periods, because now we're going to get into the real sleep factor stuff. Yeah, Gabe?
Gabriel now, University of Colorado. How in those studies, how were they sleep depriving their participants?
They would use the question was, for those of you online, is in those early subjects, how did they sleep deprive their subjects? They would use auditory stimulation. They would maintain conversations with the investigators, or if the idea was to see what, in this last few cases, what happened to the EEG, they would allow the subjects to go to sleep and then wake them up by talking to them or providing an auditory stimulus. Does that answer your question?
Yeah.
It was a lot of interaction with the investigators. Other questions or comments?
For the online. I'm not currently monitoring the questions because my computer is being
Yeah.
used. So we'll take questions from virtual audience at the end.
Okay, so now let's move into the 1960s, and this is where we really start to obtain data and learn about biology and physiology that would then drive us into studies of sleep and immune function. So during this period early on, some individuals, primarily in Europe, became interested in the earlier studies of Ishimori and Piron. So there were a couple notable laboratories that thought, well, now we have an opportunity to try and figure out what it was in brain that was accumulating that drives subsequent sleep if you've been awake for a long period of time. And the two laboratories I want to initially draw to your attention are those of Kornmuller and Monnier. Now, the early studies and many studies since have used sleep deprivation as the approach, right? You deprive the animal, you keep the animal awake, you sample your tissues or whatever, and then see if you can detect what it is that might be driving subsequent sleep. Now, both Kornmuller and Monnier took a different approach. They stimulated a region of the brain, the thalamus, that induces non-REM sleep. So they essentially put animals to sleep rather than deprive them of sleep, and then collected their tissues and samples to see if they could detect what had accumulated. So a completely different approach, but fundamentally the same kind of framework. We'll make an animal sleep more than it normally would. We'll take a sample and we'll administer those samples into recipient animals that have not been manipulated. Okay, everybody with me? Okay, before we start talking about sleep factors or somnogenic substances, we have to have a framework within which to work. So I jumped ahead just a little bit. We're still going to talk about the 60s, but by the 1980s, it was clear that we had to have some criteria so we had to have some criteria by which we could designate a substance a sleep factor. The first published set of criteria were written by Alex Borbier and Irene Tobler at the University of Zurich. And they postulated that 6 criteria must be met in order for a substance to be considered a sleep factor. By this time, sleep factor was the phrase that was being used to describe these types of research projects. And I've listed them for you here. I'm not going to read through all of them, but the important one is that the substance had to be endogenous. Right, so we're looking for actions of substances normally found in brain as opposed to pharmacologic studies where we would administer drugs and see what happened. Now, these criteria have been refined over the years, but fundamentally, they remain the same. All right, so now we're back to our sleep factor research, and this 20-year period saw an unbelievable explosion in the number of substances that were proposed as being sleep factors. So I mentioned Monnier before. He was stimulating the thalamus, extracting blood dialysates from those animals, experimental animals, and injecting them into rested animals. After a large series of experiments and I think about 6 or 8 publications, the first sleep factor was positively identified and named delta sleep inducing peptide. And it was named delta sleep inducing peptide because when administered to animals, it not only caused more behavioral sleep, but it induced these supernormal slow waves. It increased activity in the delta frequency band of the EEG, that slow wave component. Hence the name delta sleep inducing peptide. So this was the first sleep factor that met all of those criteria that Borbier and Tobler had proposed. So now we're moving into the area where we get more focused on research that led to sleep and immune interactions. Pappenheimer was a respiratory physiologist at Harvard. He had a model in which he could sleep deprive goats, and he would extract cerebral spinal fluid from those animals and focus on properties of respiratory control. He came across the studies of Ishimori and Perron, and also some of the early publications from Kornmuller and Monnier, and realized that he had a perfect model to address this question as well. So he sleep deprived goats, extracted cerebral spinal fluid from those animals, and then injected that cerebral spinal fluid into initially rats, but subsequently rabbits and other animals. So now we've already talked about dogs. We've got dog in the title. Now we're up to the goat in the title. And in these studies, what Pappenheimer demonstrated was that when cerebral spinal fluid from sleep-deprived goats was injected ICV into rats, they became less active, their locomotor activity was reduced, and they slept more. And this effect lasted for about 12 hours in this preparation and that protocol. So this paper was published in 1967. This is the demonstration of the phenomenon, the observation of the phenomenon. It took 15 years to determine what this substance might be. And by this time, Pappenheimer and his group were calling this substance factor S. So now the title of the talk is complete. We've had dogs, goats, and factor S. But there were many things they still didn't know about factor S. They actually didn't know what it was until this paper was published. This is a figure from this paper, and by now, it's an extremely interesting story, which I don't have time to tell you. They were extracting factor S from rabbit brains that were sleep deprived, and it was hard to get enough material. They also started collecting urine from medical students at Harvard. And this paper, because every metabolite in the body can be found in urine, hence their focus on urine because they had an endless supply. If you want an interesting story about how that collection process worked, take a look at the methods section of this paper. So they knew when they administered urinary factor S into experimental animals, there were periods of time when there were dramatic increases in slow wave sleep. And not only were there increases in slow wave sleep, but the amplitude of the EEG increased as well. Again, these supernormal slow waves. Jim Kruger was a postdoc in the lab. And at this time, factor S, I should back up one slide. Manfred Karnowski was a biochemist who was collaborating with Pappenheimer and Kruger. And Manfred was the one who actually determined that factor S was a compound called maramylpeptide. And this shocked everyone because there are no mammalian synthetic pathways for maramylpeptides. Maramylpeptides are components of bacterial cell walls. And the shock was, what is a bacterial cell wall doing either in brain, a component either in brain or in the urine that drives these sleep changes? So at this time, immunologists knew that maramylpeptide was a very strong immune adjuvant. They actually used maramylpeptide to drive immune responses when they wanted to study various aspects of the immune system. They also knew that maramylpeptide induced the synthesis and secretion of a substance called endogenous pyrogen, which we now know as the cytokine interleukin 1. So Kruger's logic was if MDP, maramyldipeptide, induces sleep, or maramylpeptides induce sleep, and they also induce IL-1, does IL-1 induce sleep? So this is the first full-length paper demonstrating the effects of IL-1 administration on sleep of rabbits. In those days, you couldn't just run out and buy nice lyophilized samples of IL-1. So they used different doses and volumes of a concentrate. They demonstrated that there were dose-related increases in the amount of time spent in slow wave sleep. Remember, one of our criteria, there should be dose-related changes in sleep based on increasing changes in the substance, and that there were dose-related febrile responses. Now, the fact that in most cases, if you stimulate immune activity or immune actions, you often generate or induce a febrile response. Topic for another day. Fever is a very important immune defense. So one of the criticisms that Jim heard over and over again was that your changes in sleep are simply a byproduct of the febrile response. And they have nothing to do with direct regulatory control mechanisms. It's just a byproduct. So in those days, it was kind of crude, but it was an effective experiment. And that's what's depicted in the top panel here. If rabbits were pre-treated with anisomycin, a protein synthesis inhibitor, the febrile response to IL-1 could be completely blocked, but the sleep effects of IL-1 would still be manifest. So this was the first study he did to separate the effects of thermoregulation and fever from those of sleep when activating various properties of the immune system. The same year, Irene Tobler published a paper in collaboration with Adriana Fontana, immunologist at the University of Zurich. Fontana had been working on endogenous pyrogen / IL-1 from astrocytes. So Irene was the sleep expert. So they administered some of their IL-1 preparation into rats and in this initial study, didn't observe any change in the amount of time spent in non-REM or slow wave sleep, but did describe a reduction in the amount of time spent in REM sleep. And we'll get back to the REM sleep story a little bit later. But again, the effects of IL-1 on sleep included an increase in EEG slow wave activity, or those supernormal slow waves. All right, so everybody with me thus far on these early studies that went from we're going to sleep deprive an animal and we get an increase in substance, we don't know what it is, to the point where there were some sleep factors that had been identified and characterized, notably DSIP at this point, and now factor S, which turned out to be a maramopeptide. Okay, so in the space of about 10 years, from 1980 to 1990, more and more laboratories became interested in sleep factor research, to the point where specific workshops and symposia were being organized to focus on this topic. So the first specific workshop that I could find reference to was organized by Borbier and Riadine Urson in conjunction with a sleep meeting in 1982. They had a small workshop. There were 6 presentations, and they focused on 4 purported sleep factors. Before the end of the decade, actually only 6 years later, there was a large workshop organized by Kruger and Inoue. As an aside, I joined Kruger's lab in 87. They had this workshop for a week in Hawaii in 88, and I didn't get to go. But the point is, by this time, the number of attendees at a sleep factor workshop had increased dramatically. There are more than 40 attendees, and there are a lot of famous people in here in the field of sleep. We can have some fun later and point out pictures of people, but let me, here's Emmanuel Mignot. Well, let me see. This is Sespuglio. Where's Kruger? He's there. No, I can't see him. Eric Smith, he actually recruited me to my first faculty position. So we can go through this picture and have fun later. The point I'm trying to make is that within a relatively short period of time, interest in this aspect of sleep research really took off. Books were being published as a result of these workshops. If you want a very early review, this review paper by Borbier and Tobler is excellent. So I recommend that if you want to go back in time. So if we move ahead to the 1980s, we were kind of beyond or getting beyond the point where there was a lot of skepticism. There were too many laboratories working on aspects of sleep and immune function to say you contaminated your samples, because everybody knows the brain is an immune privileged site, so there can't possibly be cytokines or immunomodulators in the brain. So we were getting beyond that point. During this sort of 20 to 30-year period, a lot of studies focused on cytokines in sleep, and I'll show you some data from that. We began to develop tools that would allow us to selectively target these systems. Platforms became available and methods became available that we could determine changes in message and protein, and then start to build more comprehensive pictures as to how components of the immune system could interact with sleep regulatory mechanisms to drive physiologic sleep, but also the changes in sleep that occur through pathology. So here's my professor emeritus unabashed picture of Kruger's lab in 1992. Here's Jim. This is his longtime collaborator from Hungary, Ferenc Obal. Linda Toth, who did the studies that we'll talk about in a moment on infection and sleep. And then you might recognize another individual in there or not. The point I'm making here, there's also a second point I'm making here. In the current climate with diversity in almost every aspect of life, our lab was extremely multicultural and multinational by accident. We brought people into the lab who we thought could contribute to the program. So we had lab members, as I was listed, from the United States, Hungary, Italy, Germany, Japan, China, and Russia. It made for a lot of fun. We had great parties. We learned a lot about each other's culture. We were able to visit other people's countries because now we have friends and colleagues. So I want to focus on two aspects of the research that was being done in Kruger's lab during this time. One was infection with replicating pathogens and how that impacted sleep. And actually, I'm not, in the interest of time, going to speak very much about that focus of research in the Kruger lab. We will spend some time talking about the effects on sleep when these immunomodulators were either administered to animals or if we targeted their systems in an animal to determine what would happen to sleep. All right, so to give you a summary slide for systematic studies that have been conducted with respect to true infections, which means a replicating pathogen, and its impact on sleep. There have been a lot of models that have been used, and a lot of infectious agents have been studied with respect to sleep. And there are many reviews on infection and sleep that are very interesting if you want to pursue them. One question about infection and sleep that I want to spend just a couple minutes on is whether or not the way sleep is changed or altered through the course of an infectious challenge, whether that is of functional consequence. In other words, the way we sleep when we're sick is much different than the way we sleep when we're healthy. Do those changes in sleep facilitate recovery from the infectious process? This is still an open question. There is evidence that it might actually be the case, particularly within the cardiovascular literature. The way you sleep after a heart attack actually determines whether or not you're going to survive or the extent of the pathologies that ensue. There haven't been a lot of systematic studies to address this question, but I want to show you one early study that gives a suggestive answer. So Linda Toth was the first person to do systematic studies of the impact of infectious agents on sleep, initially in rabbits, subsequently with a lot of work with mice. By 1993, she had published already a series of papers with the impact of gram-negative, gram-positive bacteria on sleep, and a gram-positive fungus on sleep. Now, what I'm showing you in this figure from her paper is a cumulative score that she derived called the sleep quality score. And it was simply the product of time spent in slow wave sleep and amplitude of slow waves in the delta frequency band. So she could come up with a single metric to describe sleep quality just simply based on those two. Is linear, and the solid line represents the line of identity, right? So control conditions are this first 24-hour period, and we get a cumulative increase in this sleep quality score across that time. Then at this asterisk symbols, the same animals were inoculated with these respective infectious agents, and she continued to record and determine impact on sleep. So what you notice is that some animals had, quote, good quality sleep because their sleep quality score is above the line of identity. And some animals would have poorer quality sleep because their sleep quality score was below the line of identity. Does that make sense? So when she retrospectively looked at the outcome, what she discovered was animals with good quality sleep survived. Animals with poor quality sleep were more likely to die. So as I mentioned, these data suggest that the manner in which we sleep through an infectious challenge can play a role in recovery processes. Does that make sense? Question?
What are the physiological factors which determine the quality of sleep differing, even though they're infected by the same organism?
The question is, what are the physiological factors that determine sleep quality, different sleep qualities in a group of animals infected with the same agent? All animals are different. Genetically identical mice are not identical in the way they respond to stimulus, for example. That's why we have to have a sample size. If every animal was the same in all aspects, you could do everything you needed to do with the sample size of one. So animals respond differently to the extent of the fever and other changes in systems that drive mostly, as we'll see, the arousal promoting systems. So there's biologic variability, and all that is manifest in different, quote, qualities of sleep. Does that help?
Yeah.
So if you and I are subjected to the same pathogen, we'll respond differently for a variety of reasons.
Was the age of the organism of the animals?
No. No. So I'm trying to remember, most of the rabbits that we worked with at that time were around 6 to 8 months of age. So the same way we would work in our animal studies, we try to stay within a age window unless we're doing an aging study. Any other questions or comments? Yes.
What led from like the transition to like rabbits to like rats or like goats? Was it just kind of the feasibility of using that model organism at the time, or was there something else?
So larger animals have, the question is why transition from goats and rabbits to mice and rats in these kinds of studies? Larger animals have the benefit of being easier to instrument. If you need to obtain material, you're going to get more material from a rabbit brain than you will from a mouse brain. Part of it is financial. The cost of an experimental New Zealand white rabbit is a lot different than a C57 Black 6 mouse. And over the decades, as we refine our approach to the use of animals in research, that also drives, selection of your study subject. Now, with the ease that we can manipulate the genome of the mouse, for many studies, it's the animal of choice because we can tweak things. And we'll hear from Paul on Wednesday, an even easier model to tweak things. So I think those are some reasons why. When I joined Jim's lab, I was working with rabbits, and I can tell you it's not fun to work with a rabbit. Anyway, that's a coffee break story. Okay, so by now I'm going to shift gears to the other aspect of research that was ongoing in Jim's lab, and that was a focus on actions of immunomodulators in the brain. Now, early on, when Jim and Pappenheimer and Karnofsky were first publishing that factor S is a bacterial cell wall component, the criticism was, well, you contaminated your sample. I mean, that's impossible. So you messed up. And it took a long time to convince reviewers of manuscripts and grant applications that no, you didn't contaminate your sample. This is a real thing. So by later in the 80s and the 90s, it was pretty clear that the cytokines and other immunomodulators were in brain normally, and that the machinery for them to function in a variety of processes was there normally in an animal that's not sick, or you haven't taken samples from, so you certainly didn't contaminate a sample. This is just an image of a neuron expressing IL-1 receptor type 1 in red. So this is an image from my lab 15 years or so ago. Receptors are there, the ligands are there, everything is there. So we now know, and I'm not going to go into a lot of the definitions of cytokines and how they actually work because this is a higher level type of talk, but components of the immune system called cytokines are critical for communication between and among various components of the immune system. They also function in brain, and we'll look at some of the substrates where this occurs to drive a lot of behavioral and physiological processes. Now it's well accepted that cytokines and other immunomodulators are involved in the regulation of physiological sleep, physiological body temperature or thermoregulation, locomotor activity, mood, sexual activity, appetite. So many physiologic processes driven, let's say, by regulatory mechanisms in the hypothalamus. Cytokines and immunomodulators are implicated in those physiological, i.e., normal processes, which means that they are in a place to actually function and respond very quickly to immune challenge, because all you have to do is amplify processes that are already underlying normal physiology. So I'm just briefly on 2 slides going to give high-level statements about the extent to which IL-1 and another cytokine, tumor necrosis factor, are involved in the regulation of sleep. Harvey Modosky published in the mid-80s that IL-1 concentrations in human plasma peak at sleep onset, and that in cat they vary with sleep-wake phase. Jim demonstrated in the late 80s that IL-1 and TNF message and protein exhibit a diurnal variation. Again, another criteria met for this proposed sleep factor criteria. And if you administer IL-1 or TNF to laboratory animals, their sleep is altered. Now, it's this latter point that fascinated me, in the sense that if you administer an immunomodulator, you're triggering a cascade of responses across multiple systems. So just because you administer a cytokine of choice and sleep is altered, it may be the case that it's not a direct effect, but that it's a response to something that happens downstream. So initially, we didn't have a lot of reagents, so we could give stuff, but it became apparent that we also needed to block these systems in brain. And eventually, through a period of years, a variety of reagents were produced that allowed us to selectively target these systems in brain. So antibodies began to be produced, receptor antagonists began to be produced, and of course, eventually we could just manipulate the genome of the animal and alter the systems of interest in that way. In each of these cases, if we targeted the signaling receptors for these immunomodulators, we could reduce sleep or reduce the responses to those immunomodulators. So if you pre-treat with an antibody or a receptor antagonist, and then give your cytokine of choice, IL-1 or TNF or IL-6, we can block those responses. So now we had a way to target the systems in otherwise healthy animals and do that in a very selective manner and determine effects on sleep. So by 2000-ish, 2001, we knew a lot about the interactions of these immunomodulators or cytokines, IL-1 and TNF, with other growth factors, other endocrine substances, and how that could drive changes in non-REM sleep. And this is a seminal figure from a paper that Jim published that gave an idea of the complexity of these interactions. One thing we did not know at this time, and does not appear on this figure, are the substrates in brain where these interactions are actually occurring and how they're actually being mediated. So for the next 10 years, we started a series of studies aimed at identifying the underlying mechanisms whereby a cytokine such as IL-1 would alter sleep or be involved in regulatory processes of sleep. And I better pick up the pace and try and summarize some of these in the next few minutes. And so I told you that IL-1 alters sleep of animals. IL-1 does two things to sleep if you give it to a laboratory animal. It increases non-REM sleep, and in a few minutes we'll come back to REM sleep suppression. So these are 3 different rat strains. We're not going to talk about why there are differences in time course, but in each case, there are periods following IL-1 administration where non-REM sleep is essentially doubled. We wanted to know why and how. So my colleague Luca Ameri at the University of Milan and I started a collaboration with 2 professors at UCLA, Dennis McGinty and Ron Smuziak. They had been studying the preoptic area of the hypothalamus for many years and had a really good handle on the neuronal types and subtypes in this region that were involved in regulating non-REM sleep. So in our collaboration with them, the first study we did was a very simple one: administer vehicle or IL-1 to a laboratory rat, determine the amount of sleep in the next 2 hours, sacrifice the animal, do some histologic studies to determine the number of cells in this preoptic area that were activated as evidenced by expression of CFOS. So here you see the data. After IL-1, animals slept more. Here we are, 40, 50, 60% of the time of that 2-hour period in non-REM sleep. And the number of FOS reactive neurons increased as well. So we had this association, and that's all it was, a correlation. More sleep after IL-1s associated with more cells in this brain region expressing FOS. Now, Dennis and Ron knew that in this part of the brain, there were sleep active cells, wake active cells, and state indifferent cells, neurons that did not change their firing rates through different stages of sleep. CFOS doesn't tell us what those cells are. It just says that cell was activated, but it doesn't tell us what the type was. So we did, in collaboration with them, another study where they used a technique that they had perfected where they would implant a microdialysis probe into this region. And surrounding that microdialysis probe was a microwire bundle to record activity of the cells adjacent to the probe. Then you could perfuse through the probe either artificial CSF as a control or substance of interest, in this case for us, IL-1. Now, I mentioned that they had described wake-related neurons and sleep-related neurons. The definition is very simple. Neurons that have a higher discharge rate during wakefulness are your wake-related neurons. Neurons that have a higher discharge rate during non-REM sleep than during waking are your sleep-related neurons. Our hypothesis was that IL-1 would increase non-REM sleep by increasing firing rates of sleep-related neurons. I mean, logically, that's a good hypothesis. As is often the case, the data did not support that hypothesis. Rather, they demonstrated that IL-1 in this brain region acts by suppressing or reducing firing rates of wake active neurons. So still a meaningful observation, but not what we initially hypothesized. Well, there are other parts of the brain that are involved in the regulation of non-REM sleep. One is the dorsal raphe. In conjunction with Luca's lab, we did a study where we injected IL-1 into the dorsal raphe, where these serotonergic cell bodies are located. We did a dose-related study. These data are presented as differences from vehicle control. So at the higher dose we used, we increased non-REM sleep when we microinjected into the dorsal raphe nucleus, and we also increased body temperature. REM sleep was suppressed. I'm going to step quickly through a couple slice electrophysiology slides because they show directly that when IL-1 is in the bath, firing rates are reduced of these identified serotonergic neurons. And now I'm going to get back to REM sleep. One of the most consistent findings with IL-1 administration is that REM sleep is suppressed. Well, REM sleep is regulated by different brain regions than non-REM sleep. So we went to the REM sleep area of the brain, which is really the brainstem, and I'm oversimplifying. This cluster of neurons in the lateral dorsal tegmental nucleus has been implicated by many in the regulation of REM sleep. When we microinjected IL-1 into this brain region, REM sleep was suppressed when we hit the target, but if we missed the target, there was no effect on REM sleep or non-REM sleep. So there was specificity to what we were doing. It's not just a global response. Again, stepping through slice electrophysiology studies rather quickly, these cholinergic neurons in the LDT would reduce their firing rates when IL-1 was present. So the picture that developed was one of a circuit where it involved the brainstem, the preoptic area of the hypothalamus, the HPA axis, which I haven't talked about those data, and other substances. I think in the interest of time, I want to remind you that neurons are not the only cells in brain, that we also have two other very important cell types, glia, which are astrocytes, and microglia. Early studies by many individuals demonstrated the importance of what we know now as a tripartite synapse, a synapse between two neurons, but also astrocytes and microglia. All right, I'll just quickly go. When Ashley Ngiosi, who's now an assistant professor at Ohio State, was a graduate student in the lab, she was very much interested in which component of this tripartite synapse, astrocytes or neurons, was critical for mediating IL-1 responses. So she created mice that contain the IL-1 signaling receptor. It's IL-1 receptor type 1 only on neurons and nowhere else in the brain or the body, or another set of animals that expressed IL-1 signaling receptor only on astrocytes and nowhere else. So now we had animals with selective expression of IL-1 receptors, which we could then probe to see how they responded to stimulus. So to make a longer story shorter, animals that express IL-1 receptor 1 only on neurons or only on astrocytes do not respond to IL-1 administration at all. So you notice these data, you don't even need to do statistical analyses, there's nothing there. Whereas wild type animals demonstrate REM suppression and non-REM sleep enhancement. So what this told us is that within the context of IL-1 actions, you needed that complete tripartite synapse. Because if you only have one component that's active, you don't get signaling or the response. Now, in contrast, those same animals, when it's subjected to a different kind of immune challenge, lipopolysaccharide, LPS is a bacterial cell wall component that induces an extremely strong immune response. In this case, it was sufficient to have IL-1 receptors either on neurons or on astrocytes to get the response. So in this case, we've demonstrated that depending on the type of immune challenge, the cell types on which these IL-1 receptors reside may be more or less important in mediating sleep responses to that challenge. And I think I'm going to stop there because I'm at the end of my time. I actually thought 75 minutes would be a really long talk, but it seems like Ken knew better. So some high level conclusions, and I want to leave this slide up. Much of what I've told you today is published in this paper that just came out a few months ago. And this paper is part of a Festschrift in honor of Jim Kruger. For those of you who might not know, I put the definition of a Festschrift. It's a collection of papers written to honor a scholar. So this is in honor of the contributions that Jim Kruger made to the field of sleep and immune studies. Those two, this QR code is for the whole series of papers in that collection. And this one on the left is for the review paper that I wrote. So thank you very much for your attention and for learning a little bit about history.
So thank you very much, Mark. Don't leave yet, because we are going to transition to a coffee break, but we are going to have a few minutes for questions and answers here. So one of the things I wanted to say that I didn't mention is I'm not going to give detailed introductions to all the faculty here because I want to make sure they have time to give their talks, which worked out. But I wanted to say that they're all leading experts, world experts in their field, as you just saw. All right. And so that's for all the faculty who are participating here today. So thank you. All right. So questions. We got one question here. Sharon from Johns Hopkins. Are we assuming all sleep-related neurons are sleep promoting? Shaheen, sorry.
It's a fine distinction, and it might be difficult in some cases to determine whether sleep-related and sleep promoting are the same or different. So obviously, if you think about, use an analogy of a correlation, you can correlate alcoholism with the number of Baptist preachers in a town, but it doesn't mean that they're directly related. And I think that's the way we have to focus on this topic. Now, we know that most sleep-related neurons are also warm sensitive, so there are other thermoregulatory aspects to consider. But to make the distinction, I think a sleep promoting neuron will change its activity and then behavior will change, right? So I change my firing rate if I'm a sleep promoting neuron, and the behavioral manifestation or some manifestation will change. If I change my behavior and other neurons then have altered firing rates, that could be simply a related phenomenon, but not necessarily. So the short answer is no, I don't think we're assuming that all sleep-related neurons are also sleep promoting.
Questions from either the virtual or in-person audience?
Hi, Keenan Kushner, to you, Boulder. I'm just curious on the IL-1 receptor, what exactly do you believe is happening that is allowing it to suppress those serotonergic neurons?
There are a lot.
So Mark, repeat. Thanks.
The question is, what's happening with the IL-1 receptors to suppress activity of the serotonergic neurons? Those are GABA-mediated effects. And a card-carrying electrophysiologist could go into a lot more painstaking detail than I could.
Hi, my name is Allison from University of Puerto Rico. My question is, considering the history of sleep research, how is it that despite everything we know about sleep and sometimes medical trainings remain among the most sleep deprived, and then what barriers prevent us from resolving that paradox?
Did you get that?
Yeah.
So if I understand, the question is, given what we know about sleep and the health consequences of poor sleep, why is it that medical students and interns and residents are so deprived of sleep? Well, I've never gone to medical school, but I can tell you based on studies that have come out of Ken's mentors group, that if you change the shift schedules so more sleep is available or more sleep opportunities are available, you reduce the number of medical errors. So that seems fairly clear. But it's not the type of data that wants to be accepted. And I think a lot of this is driven by well, this is the way we've always done it, so we need to continue training in that same vein, or an answer that's intended to be more quantitative is, if you're a resident, you need to see a certain number of cases in order to gain the skill sets that you need. And if you're not on call, you won't see the number of cases that you need to obtain. And Ken probably has a lot of other variations on that kind of theme, but.
Nate Anderson from City of Boulder. First of all, great talk, thank you. Two things. First, a comment. Also a fan of history. The doctor that invented the modern residency schedule was an advocate of using cocaine at that time and throughout your career. So I think everyone should know that. Maybe make adjustments accordingly.
But and also to relate to that, residency meant you lived there,
Yeah.
you followed the progress of the disease, and a lot of them back then the patients were dying.
Yeah.
So you were there for a couple days. You weren't there for months. You know, that kind of stuff. Yeah. So yes, there are many factors there that tap into that. Yeah.
And then if I can follow with a question, I'm curious about some of the methods when you were doing some of the early work here. How did you know the difference between injecting your particular peptide of interest versus injecting anything that caused an immune response?
So the question really deals about controls for the study. Obviously, if you implant chronic guide cannula into the ventricular system, so you can administer substances ICV, the fact that you penetrate the tissue causes damage, of course, and a localized inflammatory response will occur. So you control for that by implanting all your animals, allowing them sufficient time to recover. You can never eliminate entirely those unanticipated consequences, but you can control for them in how you set up your protocols. So we would initially implant the guide cannula, obtain recordings from animals after recovery with no manipulation at all. Then after administration of either artificial cerebrospinal fluid or saline, depending on the compound we wanted to administer. And then also, followed up with the experimental injection. So you can account for the inflammatory response to the tissue damage with a vehicle control, but you can't, not with EEG methods. It's much more difficult to determine this much change is due to the surgical procedure itself. But you have to build those controls in. Other types of controls we had to use in the early days, the systems used to express the proteins, which were then lyophilized and shipped out, used an expression system that was prone to contamination with bacterial cell wall products. So one control that we always had to do early on is heat and activate, let's say IL-1. If we were doing an IL-1 study, we would have to heat and activate it, because the heat would denature or change the conformation of the protein, and the protein would no longer have a biologic effect. But if you had a response in terms of body temperature or sleep after that denaturing of the protein, then you knew that you had contamination with a cell wall product. So for years and years and years, we had to run a heat denatured control. Go to coffee break.
What, definitely one last question here. No? Any last questions from abroad? Let's see, thank you for the explanation. All right, great. All right, well, thank you again. For our virtual audience, we're going to be back promptly and start at 10:45 Mountain Time, Colorado.